Hello my dear students. Today we will discuss about the electrochemical cells. That means voltaic cells or galvanic cells. Basically we will study the Daniell cell and its working. We have to also study about the short presentation of the cells and the last one, that is the detection of electrode potential, where we consider SHE or NHE, that is the normal hydrogen electrode or standard hydrogen electrode. So let's start the first topic. Well, you see that the first topic, that is the electrochemical cell or galvanic cell, and we also understand that those kinds of cells which can convert the chemical energy into electrical energy, right? They are those devices which can convert the chemical energy into what? Electrical energy. It means they are such cells where the kind of reaction is taking place inside the cell. That is basically the redox reaction, and we know that the redox reaction consisting what both oxidation and of course reduction. So basically redox reaction is going on in such cells where we get the electrode potential, right? So such cells are, you can say, galvanic cells or electrochemical cells. We can also go for the voltaic cell as well. Here we can consider the Daniell cell. Let's see the construction of the Daniell cell first. Here we go, the Daniell cell. Well, the Daniell cell, it is actually consisting two glass containers. We can mention as I'm showing in the diagram like this. There are two chambers, or you can say two containers, A and B. And in container A we have the electrolyte or electrolytic solution of zinc sulfate. So this is the zinc sulfate, and we are putting a rod. This is the zinc rod. This is zinc rod. All right, this zinc rod is further connected with the voltmeter. Okay, and at other hand, in the B container we have the copper sulfate solution. Here we have the copper sulfate solution. You know that the color of the copper sulfate solution, that is blue. So copper sulfate solution is here, and I'm putting a rod that will be made of copper. So it's a copper rod. Both are connected with a voltmeter, as you can see. Okay, so these are the two chambers. There is one more thing which is very important, which is being called a salt bridge. You see that the hard glass U-tube I'm showing here like this. Here both ends are actually filled with cotton gauze, and in this U-tube we have the electrolytic solution of potassium chloride. This is the salt bridge. So salt bridge consists of what? KCl, the potassium chloride electrolytic solution, and of course one more solution is there, that is agar. Okay, so it's been sealed at both of the ends. So basically this is the Daniell cell for you. Here you can say that the zinc rod, it actually works as anode, and it's a Daniell cell or electrochemical cell. That's why it has been negatively charged portion. And at the other hand, we have the copper rod. It works as what, cathode, and it works or it's charged, you can say. Electrode is positive charge, I mean to say. Alright, so this is the basic construction of the Daniell cell. Now we move on to the kind of, you can say, reaction which is taking place at each and every, you can say, electrode. Let's see one by one. First of all, we are talking about the anode reaction. That means reaction at anode. Alright, so at anode you have to mention the oxidation reaction basically. Alright, so what is oxidation? It's a removal of electron. So basically what is going on here, the zinc is turned up into Zn2 plus and 2 electrons are released, and these 2 electrons are released no doubt in the solution. Okay, and this is the oxidation reaction. Whereas at other hand we have cathode, so we can go for the reaction at cathode, and it will be the copper 2 plus ions, right? So CuSO4 is split into Cu2 plus ions and SO4 2 negative ions. So basically Cu2 plus ions, they're actually catching 2 electrons which are released from anode, right? And obviously it will turn up into what, copper, right? So addition of electron, that is reduction. So we can write here the reduction reaction. When we consider both of the reaction where the number of electrons are gathered here, that's why it's been the negative charge, and these electrons are going from this end to this end. So obviously by the kind of, you can say, redox reaction which is taking place in the cell, okay, we get the electrode potential value that comes what, E equal to 1.10 volt. That will be detected by the voltmeter. So the kind of, you can say, redox reaction taking place here, we are just adding both of the equation here. Then you see that the 2 electrons and 2 electrons being cancelled, then the left hand portion, that is the zinc and Cu2 plus, they actually gives what, Zn2 plus and Cu. Okay, so these are the half cell reactions which are taking place at anode and of course cathode, and this is the complete, you can say, redox reaction, or you can say the net cell reaction which is undergo in this Daniell cell. Okay, so this is the working of Daniell cell, and one question arise here, that is what is salt base? The salt base is actually breezing both of the, you can say, half cells basically, and it's very important thing. Without, you can say, salt bridge, this cell never works. Okay, so salt bridge is made of hard glass tube, where you can say the electrolytic solution, potassium chloride is there, and the agar solution, and both ends of the tube is sealed by the cotton gauze. So exactly the function of the salt bridge, the very first one, that is very important. It can basically stop the intermixing of the solution. It means what, this solution never goes from this chamber to this chamber or this chamber to this chamber, right? So basically it can stop the intermixing of the solution. First of all, the selected ions can go from one end to another end, and number two, that is the electrical neutrality. All right, so it can maintain the electrical neutrality in the cell. So these two are the very important, you can say, points as far as the salt bridge is concerned. The first one, that is, it can stop the intermixing of the solution or electrolytic solution. Number two, it can maintain the electrical neutrality as well. So this is all about the Daniell cell. Now in the next section we will study about how to represent, you can say, Daniell cell. Let's see. Well, here we have the NCR, that is the net cell reaction, or the redox reaction which is taking place in the cell. Now the short presentation or representation of the cell. First of all, what you have to do, you just make two vertical lines like this, and one horizontal line, and this left portion always considered what as anode, okay, where you can say that oxidation reaction always perform, right? Obviously it is mentioned by negative sign. At the right hand side we have to mention the cathode, and reduction reaction actually occurs here. It shows by positive sign like this, and two more lines are also there. In the first, you can say half, left hand, we can introduce the electrolyte. So we were using what, zinc sulfate here in this manner, okay. Its molarity you can write as well. So what kind of molarity we have, or concentration, and the kind of, you can say, oxidation reaction is taking place. That means the zinc turn up into Zn2 plus, right? So we can write in this manner. So zinc is turn up into what, Zn2 plus, right? So this is the right way to express the anode portion. Now we move on to the right hand side, that is the reduction. That means the cathode reaction is going on, right? Here we can use the copper sulfate solution, whose molarity that can be written as well, and here we can say the Cu2 plus 2 turn up into what, Cu. So you can write Cu2 plus 2 Cu. Alright, so this is the actual presentation of you can say the Daniell cell. So any kind of cell you can easily I mean to say write in short manner. Well, let's see that the next topic is. Now this topic is very, very important, and this topic is SHE, or you can say NHE. This is the short term of standard hydrogen electrode, or you can say normal hydrogen electrode, okay. So why we are studying this, SHE or NHE? It's a very important thing. Basically we have to find out that a particular electrode having the kind of, you can say, electrode potential value, right? So that will be only detected by using of some kind of standard electrode potential value. So there is a comparison between the unknown electrode potential with the known electrode potential value. So we can easily detect by using of SHE and NHE. All right, so later on you will get the answer that why we are why we are studying this SHE or and NHE. Well, we see first of all that the construction of SHE or NHE. I'm just again repeating that SHE is the standard hydrogen electrode and the NHE is the normal hydrogen electrode, and its electrode potential value, it is been denoted, you can say E she or not always be, it is 0.00 volt. In any case, you can say either the kind of, you can say, oxidation reaction is taking place or reduction reaction is taking place at the electrode. So basically E she value always be what? 0.00 volt, right? So first of all, let's see that what is the construction of SHE or NHE. All right, so here we are the construction of SHE or NHE. Here you can see this is the container and it is containing what? 1 mHCl, 1 molar, you can say, HCl. And this is the glass you can say tube or chamber, you can say, where the hydrogen gas is passing at 1 bar pressure. And inside we have the platinum black. Actually this platinum is a plate, right? Platinum plate we have taken, which has been fused by the concentrated HCl, right? That's why it's turned up into what? Platinum black, right? It is connecting with the platinum wire and finally it is connected with the connecting wire here. So this is the complete structure of SHE, and let's see that how it works. So as far as anode is concerned, right, so we have the reaction whenever it works as what? Anode. You see that at anode oxidation reaction is taking place and it's been the negatively charged, right? And you see that we have the hydrogen, right? And hydrogen is turned up into 2H plus. It means 2 electrons are released from it. So basically what is going on here? 2 electrons are out from this H2. That's why it is turned up into what? 2H plus. Clear? It is clearly showing that the anodic reaction where the oxidation reaction is taking place because electrons are removing. Clear? So this is the anodic reaction or anode reaction. And if it works as cathode, right, then obviously reduction reaction is taking place. This is the positive H. So what is going on here? Basically HCl is turned up into H plus and Cl negative ions like this. So H plus and Cl negative ions are splitting by the HCl. So you can say that 2H plus ions means this reaction is revised or reversed. 2H plus, actually they are gaining 2 electrons to become what? Hydrogen. Okay, so these two reactions are taking place at anode. Undoubtedly electrons are releasing, and whenever it works as what? Cathode, then the 2 electrons are consuming by 2H plus and obviously it will turn up into what? Hydrogen. So basically anode and cathode, both reactions are taking place, but depend on condition that what kind of, you can say, electrode you have, where you just comparing with this sheet. Okay, so this is the working of SHE or NHE, right? In later on I will discuss about how the SHE or NHE and other, you can say, electrode compare, and how can we detect the electrode potential of a particular electrode. All right, maybe in the next video we'll discuss about such things. Okay, so let's see that what will be the short presentation of this anode and cathode reaction. Well, you see that at anode, where the oxidation reaction is taking place, H2 turn up into 2H plus and 2 electrons are releasing, right? So what you have to do, you have to mention 2 lines here. This has been the salt bridge, you can say. And of course, there is the electrolytic solution, that is HCl, or 1 HCl is there. And you have to mention that H2 that will turn up into 2H plus in this manner. So this will be the anodic reaction because it's existed at the left hand side, so it's an anodic reaction. And what about the cathodic reaction? So as a cathode, all right, so the mentioned reaction that will be reverse of the anode reaction. So you can say that 2H plus and 2 electrons both are reacting to give what? Hydrogen. So you have to mention the 2 lines which is showing the bridge, alright, or separating, you can say, portion. You have to mention at the right hand side because it's the cathodic reaction. So mention the electrolyte here, the HCl, and of course this reaction where the 2H plus getting 2 electrons to form what? H2. Okay, so this is a short presentation for you for SHE or NHE at whenever it works as anode or cathode, right? So in today's video what we have studied, the first one, that is the voltaic cell or galvanic cell. Next, that is the Daniel cell, the particular example, and the short presentation of the Daniel cell, and of course SHE or energy, and of course its short presentation. So thanks for watching. We'll meet in the next video. Bye bye.